Hello, everyone, and welcome to today's webinar, Decoding the Tumor Microenvironment with Paletrra, Revolutionizing Biomarker Discovery in Clinical Trials. I am Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by NeoGenomics Laboratories Incorporated. To learn more, visit neogenomics.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. I'd like to now welcome our speakers for today's event. Dr. Kirsteen Maclean, Principal Scientist, Scientific Affairs at NeoGenomics, and Herein Nunns, Senior Scientist, Senior Manager at NeoGenomics Laboratories. Dr. Maclean, I will now pass it over to you for your portion of the presentation.
A huge thank you to LabRoots for hosting this webinar today, as well as the introduction. Again, I'm Kirsteen Maclean, and I'm the Director of Scientific Affairs within our Pharma Services Division, and I'll be joined by my colleague, Herein Nunns, who heads up our spatial image analysis, as well as some of the data analytics. And we're really excited to be presenting our new Paletrra solution within the spatial environment. And so, given that I think most people on this webinar have an interest actually in the oncology space and potential novel therapeutics and clinical trials, and also maybe opportunities for improved patient care, I'd really like to share with you our mission and vision statement, which is really centered around saving lives by improving patient care and really being one of the leading providers of comprehensive cancer testing solutions. And so what we'll be covering today really is a variety of topics, including what you see in front of you, which is really what it is that we do within Pharma Services, a brief overview of some of our validated as well as verified spatial solutions to address some of those potential changes within the tumor microenvironment. We'll also walk through some specific applications with our new Paletrra workflow, and then we'll wrap up with some case studies as well as an overview. And so fundamentally, we are a CAP/CLIA approved oncology focused company that puts patients really first and foremost. And we do this with really synergy across our clinical, our data solutions, and Pharma Services division. Again, I'm part of Pharma Services. And for the most part, this is providing up-to-date technologies, assays, and diagnostics for improved decision-making. Our data analysis division actually includes this wealth of data available to us and really can help match patients expressing specific biomarkers or mutations with appropriate trials, and also assist in some of our drug discovery and development. Now, specifically, we are very well positioned to offer this multi-modality approach to clinical trial testing, and I'll be showcasing some of the slides alluding to that. So this is a really kind of nice overview of where our different sites are today, as well as the type of testing modalities that are supported. And specifically for our spatial assays, most of the work is actually run at our site in California. And you can see we actually offer a lot of modalities within that California site. And all of that is actually supported from a very large team of pathologists. And so in 2025, from a Pharma Services perspective, we're really focusing in on offering solutions under the umbrella of what we're deeming specific therapeutic modalities. And I think we can all appreciate, and I've certainly gleaned a lot from this, being at the recent ASCO meeting, is that traditional approaches to cancer therapy, such as chemo, radiation, or surgery, have really been, they've burdened patients, certainly because they have very limited long-term responses. And so really this, I think, landscape has changed over the last few years. And again, really highlighted at the recent AACR and ASCO conferences in the US, we're really starting to see the utility in some of those promising results from using cell-based therapies, antibody-drug conjugates, and also improved small molecule target therapies. And really these cutting-edge treatments are really now what's offering a more personalized and precise tumor targeting. And they also, from some of the recent clinical trials, can really improve that overall potential to prevent disease progression. Now, on the right-hand side, you can see some of the very comprehensive portfolio of not just validated platforms, but assays that can either be ready to use for your trial needs today, or they can be customized for your specific trial needs. Now, of course, this biomarker testing includes supporting IHC, next-gen sequencing efforts, so that's under the umbrella of genomics. We have a strong fish cytogenetics team. We also include nanostring gene expression. And of course, from what we'll discuss today, is our spatial solutions. And, that's wrapped under spatial analysis, and a lot of that's handled by Herein and his team, as well as some of our pathologists that we have on site in our California testing site. And so I think it's become very clear over the last few years that the use of spatial biology and essentially spatial biology tools, which again can be both transcriptomic or proteomic for the most part, have really revolutionized our understanding of complex biological systems, especially in cancer, because really it allows that very intricate spatial relationships to be looked at between proteins and genes and cells and tissues. Now, it's a very innovative approach, and it allows not only the visualization of potential biomarkers, but also the distribution and interaction of these biomarkers, leading to improved understanding really of biology and disease, and as well as understanding mechanisms and therapeutic responses. And again, aspects of that my colleague Harry will be going through. Now, of course, despite all these advances, there are several challenges that actually hinder the full potential of spatial proteomics in the clinic today. And I've just highlighted a few of those in this slide. Clearly, there's a need for data management together with the need for appropriate computational tools, and really thinking about this more, I would say, interdisciplinary kind of collaboration approach to effectively analyze and interpret spatial data. And other challenges, and again, you can have a look at the list that I've put together. I think other challenges include this kind of lack of standardization today, as well as some of the pre-analytical variables that can obviously affect some of that interpretation. I think all of this suggests that there's a real need in spatial biology today to really think about improvement in translating spatial biology research into clinical practice and really defining proper conditions, especially within QC. And so at NeoGenomics, again, we do have that interdisciplinary approach to support several platforms and technology. And really, as part of our spatial portfolio, this means that we have a dedicated pathology team, but we also have a dedicated scientific team to the spatial biology and a dedicated data analysis and analytics specialists. So let's consider how one would integrate some of the spatial solutions into a clinical trial and what spatial solutions we actually offer at NeoGenomics. And so the geomics from NanoString, and it's now Bruker Spatial Biology, that really supports the whole and maybe cancer transcriptome, really a discovery approach where one can think about narrowing down specific pathways and targets. Then we can further triage and perform these kind of proof of concept studies and exploratory trials with the use of our new Paletrra workflow. And this really enables a more detailed look at the tumor microenvironment. And then as far as CDX, this is certainly something that we can also support, especially when looking at IHC. Now, again, if you look at left to right, that indicates really the number of samples that one would consider in the continuum of a clinical trial. And so without further ado, let me introduce you to Paletrra. This is really one of the beautiful images and one of the many, many examples that you'll see with the use of our Paletrra solution. And in this case, we're looking at non-small cell lung carcinoma. And really, it's all about the visualization and being able to tell the whole story of the tissue and using that to advance therapeutics. Now, this is a really kind of very quick kind of overview as to our overall workflow. But what do we mean by end-to-end solution? Well, it's really about the due diligence and rigorous quality control that we apply to qualifying all of our antibodies, and also we are also qualifying our ISH probes for RNA, if that is of interest to anyone on the call today. And we have that, we're able to do that because we have a dedicated team of pathologists and scientists involved in enabling this assay. Now we have our multiplexing chemistry, and this involves cyclic immunofluorescence with a Cy3, Cy5 fluorophore approach. And we also have a proprietary dye inactivation step. We acquire all these images using a rare site finder, and then we perform the data analytics that my colleague will discuss in more detail today. And so this is another beautiful example of the type of images that one can expect when running a Paletrra solution. And this time we're looking at CRC tissue, so that's colorectal tissue, wherein we can profile up to 60 biomarkers in a single FFPE section, really to help understand your overall tumor biology. Again, beautiful images that can be achieved with our solution. And so what we offer is a very unique approach to enable that detailed high resolution look at the tumor microenvironment, wherein with our Paletrra workflow, a client can either choose the pre-verified off-the-shelf panel or create their own custom panel. And here is a really nice example of really looking at various lymphocytes as well as myeloid cells. So again, top left, again, if you hone in on this particular figure, top left, is various types of T cells. Bottom left, you can look specifically at those cells expressing PD-L1, as well as highlighting some macrophages. Top right, we're looking at both T cells, B cells, and also macrophages, and also activated T cells. And then in the bottom right, we have those that express LAG3. Now that's indicative of immune exhaustion, which is also a big problem in clinical trials today. So something that I would suggest is much needed as far as anybody thinking about using spatial biology. And so my colleague Harry will actually be doing a deep dive into this, but suffice to say, our dedicated analytics, we've really specifically optimized the workflow for that. You can really enable your biomarker discovery, target validation, and mechanism of action. Now, some of the cell level as well as slide level outputs I've highlighted on this slide. I'm not going to go through every single one of them again. My colleague's going to go through that. But suffice to say, we can do specific segmentations, nearest neighbor, proximity, and also look at, who's who in the cell neighborhood, as well as some of the various intensities for these specific biomarkers. And so when you think about our image analysis pipeline, it really employs a very comprehensive three-step process. And again, it's 100% scientist-enabled QC approach. And here we utilize a virtual H&E overlay with our biomarker staining, and the AI segments that tissue to delineate out the key regions, such as, and you can see that from the figure, such as the tumor, the stroma, and also the vasculature, whilst at the same time also excluding necrosis, any issues with the tissue such as folds or also potential artifacts. And then we have the cell segmentation and obviously some of our biomarker classification. Now, as a reminder, virtual H&Es, in this case, is generated with a pseudo blend of DAPI and the autofluorescence channel. And for all our slides, in fact, all our workflows included that we'll be highlighting, we always include a pathologist to select the tumor relevant regions of the tissue. And so thinking about QC and validation, if you remember, I brought this up as a potential challenge right now in spatial biology. Well, we have a very extensive validation process, but here we actually perform concordant studies between our multiplex immunofluorescence assay and IHC. Again, that's that gold standard. And this is an example using NSCLC. And of course, to address some of the staining accuracy, correlation coefficients are actually calculated using cell density data, and that's generated with our multiplex approach and of course benchmarked to that gold standard IHC assay. And as you can see from this figure, we're staining sequential slides either with Ki67 or PDL1. Now, that top row that you see, that's our whole tissue image. Again, that whole tissue image generated with our Paletrra workflow. And the bottom is our magnified image, wherein we're looking at DAPI staining, the chromogenic, as well as the IHC. And the staining pattern is very similar across. And our concordances across the board are well over 0.94, which is excellent. Again, just a little bit more about some of our QC process that we include for all our panels to demonstrate repeatability and reproducibility of our workflow. Here we've generated data again from three NSCLC samples, and they're run in triplicate in 3 separate batches. So that's 9 total slides, and we have both inter-run and intra-run CVs calculated for cell density and as well as intensity. And again, the images that you see from these 9 serial sections are used for this specific precision study. And, you know, we're highlighting the target CD3, which is in red, CD11B, which is in green, and CD31, which is in blue, as well as the pan-CK and cyan. But this is really data that's been generated from our very specific panel that we have available as an off-the-shelf. The larger panel is actually the TME panel and includes a wealth of targets that I've listed on that second bullet point. And again, all of our CVs are less than 25%, which indicates a very, very robust assay and very robust assay performance. And so let's go back to some of those amazing images that we have. Again, this is NSCLC tissue. And again, just to recap before I hand over to my colleague, this is with the use of our new whole tissue imaging solution, our Paletrra solution that really provides that greater histological context. And really, it's about that truly unbiased spatial analysis of the tumor microenvironment. It really is able to hone in on some of that very specific detail and hone in on some of those relationships of those cells that I spoke about, and really what is their context. And again, over the next few slides, we'll be providing a lot more information as to how we can go about that with our image analysis, as well as really, in a nutshell, really about improving our therapeutic decision making. So I'm going to hand over to my colleague for some exciting updates on how we perform some of these wonderful images. Thank you.
Thanks, Kirsty. So in the next few slides, I'll walk you through the details of our image analysis process and end with a case study of non-small cell lung cancer samples. Kirsty did a great job walking through some of this. So at a high level, we perform our image analysis pipeline is broken down into 3 key steps: tissue segmentation to delineate tumor and stroma, cell segmentation, followed by biomarker classification and cell phenotyping. Coupled with this process is 100% QC of all of our stain images, including batch controls, as well as all of our analytic outputs. Once we complete analysis, we archive image files as well as any analysis settings that allow us to preserve those settings throughout the duration of a clinical trial. Now I'll dive into each of these 3 steps in a bit more detail, beginning with tissue segmentation. As Kirsty mentioned, we rely on a virtual H&E, which is a pseudo-color blend of DAPI and autofluorescence that provides histological context as we evaluate our samples, and we rely on this blend throughout our analysis process. The tissue segmentation begins with our amazing team of pathologists at NeoGenomics that annotate out all tumor-relevant regions of tissue. This ensures that during our analysis, we're capturing reactive immune content and not incorporating uninvolved normal tissue within our analysis. Within this tumor-relevant tissue, we use a combination of algorithmic and manual annotations to remove various regions that are not suitable for single-cell analysis, such as necrosis and tissue folds. Next, we segment the tissue into functionally distinct tissue regions. Here we have the virtual H&E again, showing a carcinoma with intermingled stroma throughout on the left side. In the middle, we have tumor cytokeratin staining in white with CD3 T cell staining in red. And then finally, on the right side, we have our tumor segmentation mask. And this really provides the first glimpse into the spatial distribution of key immune phenotypes, as you can distinguish intrastromal T cells, shown with a green arrow, from intratumoral T cells, shown with red arrows, that are closely interacting with the tumor cells. We also support additional biomarker and pathologist-guided region annotations, such as leading edge of the tumor and tertiary lymphoid structures. After this, we perform cell segmentation with DAPI processed by a neural network to identify each single nucleus. This is a critical step, as any errors will propagate to phenotyping inaccuracies. And we've trained our segmentation algorithm on a large library of expertly annotated nuclei across a variety of body sites, tumor indications, and cell types. And this ensures that our algorithm performs excellently as we analyze the variety of tumor samples that come in for our clinical trials that we're testing for. We then segment subcellular compartments. This allows us to quantify subcellular staining patterns. On the top panel are examples of biomarker staining, and on the bottom is the extracted single cell intensities. So as we go from left to right, we see nuclear stains, circumferential immune stains, cytoplasmic stains, and then membranous tumor stains that have been quantified through this pipeline. Final and most important step of this three-step process is the cell phenotyping. Here we consult with our pathology team for biomarker interpretation and train neural networks on millions of single-cell annotations to integrate intensity alongside staining pattern and cell morphology. And by encoding pathologist guidelines into our algorithms, we achieve strong concordance with gold standard IHC assays. From biomarker classification, we generate cell phenotypes by looking at co-expressions of multiple markers. As examples, here are CD3, CD4 T helper cells shown with the yellow arrow, and then below is a CD3, CD4 FOXP3 positive cell indicating a T regulatory cell. For many of our multiplexed panels, there may be dozens of relevant co-expressions that can be produced from our data. We report out the cell counts and densities for each of the individual biomarkers, as well as for all of the co-expressions. Now, for heterogeneous tissue specimens, cell prevalence doesn't always capture the full complexity of the tumor microenvironment. And to this end, we offer additional advanced and spatial analytics that can be tailored to each specific project. I'll walk briefly through 6 of these outputs. So to start with, region analysis, I discussed this before. This allows us to quantify cell prevalence within distinct tissue regions. In addition, intensity metrics allow us to quantify the expression levels of modulated biomarkers. And then nearest neighbor distances allow us to understand the proximity and relationship between 2 cell types of interest. Then biomarker profiling, as shown in the panel 4 with the yellow receptors indicating a biomarker of interest, allow us to understand both the distribution of a biomarker across cell types, for instance, across this purple cell type and the cyan cell type, as well as the prevalence within each specific cell type for really an in-depth exploration into where a biomarker of interest is expressed in our tissue samples. Then proximity allows us to interrogate the cell composition within a specified radius of target cells, allowing us to identify co-occurring cell types. And similarly, unsupervised clustering of cell neighborhoods allows us to identify recurring cell neighborhood motifs within tissue specimens. So next, I'll go through some of the data we generated for a batch of 19 non-small cell lung resections that were stained with a 16-plex panel in our lab. And so on the left side here, we have a heat map showing the z-score normalized densities of all the key biomarkers and co-expressions for these 19 samples. And at a high level, this allows us to see samples with high expression, or for instance, hot immune samples in red, and compare them and stratify them against cold samples in blue. And overall, through this analysis, we identified and profiled over 8 million cells, with the plurality being tumor cells, followed by fibroblasts and T cells. But again, taking these numbers in aggregate loses some of the important spatial context of these tumors. So let's take a deeper dive into 3 of these samples, which cover the range of phenotypes from immune infiltrated to immune excluded to cold immune. I'll start with a T cell infiltrated tumor, which has high T cell abundance within the tumor and tumor stromal interface, shown on the left overlay in green and blue. We also see throughout this sample PD-L1 positive tumor in orange and PD-L1 negative tumor in yellow. Overall, as we look at the PD-1 expressing T cells in this sample with the upper middle panels, we can see that they're closely associated with their PD-L1 ligand, with these T cells being within 50 microns of both PD-L1 positive tumor as well as PD-L1 positive macrophages, shown in orange and green in these kernel density estimate plots. This is in accordance with the figure on the right, top right, which shows the tumor proportion score and combined tumor and immune positivity of PD-L1 for this sample, which indicates a highly immunosuppressive environment. As we dive a little bit deeper into the T cell subtypes identified within this sample in the bottom left bar plot, we can see that the predominant subtype is the cytotoxic T cell, with PD-1 expression, Ki67, and granzyme B expression observed in a subset of these cytotoxic T cells. Then as we move to the bottom middle figure, we can see that of those cytotoxic T cells, the density is highest within the stroma, but there is high infiltration of these T cells into the tumor parenchyma as you move from the stroma on the left to the tumor on the right. And then finally, in the bottom right is a radar plot showing the prevalence of various cell types within the sample, from T cell subtypes, myeloid subtypes, endothelial cells, and cancer-associated fibroblasts. The next sample is an immune-excluded phenotype with a dramatically different profile that isn't captured within the overall cell abundance. The majority of the T cells are found far from the tumor in blue, and also in myeloid-enriched neighborhoods in purple. These T cells are not interacting closely with PD-L1, and overall there's lower but positive PD-L1 expression within this sample. As we look deeper into the T cell subtypes, we see that T helper cells are most abundant, followed by T regulatory cells, with very little infiltration directly into the tumor parenchyma. And finally, in the bottom right, we can see that the predominant cell types within the tumor microenvironment are cancer-associated fibroblasts and myeloid-derived suppressor cells, both of which may play a role in immune exclusion for this sample. And then finally, here's a cold sample with very little T cell involvement and negative PD-L1 status. Of the T cells that are present, you can see that the predominant T cell subtype is T regulatory cell. And then as we move to the radar plot in the bottom right, the predominant cell types within the tumor microenvironment are fibroblasts again and macrophages. To summarize, I've examined 3 samples within this data set of 19 tumor specimens that demonstrate the use of combined cell profiling and spatial analytics that can uncover therapeutically relevant characteristics of these patient samples. And finally, I'll play a brief video that provides an overview of this end-to-end analysis process that I've just gone over. So we begin with the virtual H&E and then toggle to a biomarker overlay that shows whole tissue context of the staining. And as we zoom in, we begin to see the subcellular staining patterns emerge. We apply cell phenotyping, and then we uncover cell-cell interactions and spatial distributions of all of the key immune cells within this tissue. And now I will pass it back to Kirsteen. Thanks for your time.
Thanks, Harry. Harry and his team really make our image analysis and analytics look very, very easy. But believe me, there's a lot of work that goes into this. And so again, kudos to his team for really developing some of these analytics specifically for our Paletrra workflow. So as we kind of wrap up, I just want to highlight a few very recent case studies really showcasing how our spatial services have enabled insights, especially into early phase clinical trials. And again, this has been really because of our dedicated team that have been running these type of assays for well over 10 years. So we have a wealth of experience actually in spatial biology. And I just want to highlight a few of those. Again, thinking about left to right, BioNTech actually recently, earlier this year, had a really nice paper in Nature Medicine where they're characterizing their specific neoantigen-specific autologous T cell product and looking in patients that either had advanced or metastatic melanoma. And you can actually see there's a really nice harmonization in this, what I've highlighted between the tumor imaging, as well as a more detailed look using spatial biology as to why, and maybe some of the mechanisms of why that tumor is actually shrinking. GSK, you can see there in the middle, analyzed some of our baseline and week 6 tumor biopsy samples, and really to show, the presence of some of that immune infiltrates, and looked to see that there was actually decreased Tregs. But on the flip side, could actually see an increase in actually NK cells, which was very nice. And then most recently, again, also this year, Infinity Therapeutics, really as part of their Mario 3 trial, which looks at patients actually with metastatic triple negative breast cancer, and really characterized some of those immune subsets again, in paired tumor biopsies. And again, the patients on that trial, it was a combination of really a macrophage inhibitor together with a PD-L1 inhibitor. But again, really nice showcasing of our spatial solutions and our dedicated team at NeoGenomics that support these particular studies. And so, again, I've talked about some of our off-the-shelf products that we have as far as Paletrra, and really enabling you to get up to speed very quickly with addressing some of the needs for your clinical trial. But this is a very evolving test menu. And as I mentioned, we have several what we deem off the shelf. These have gone through a lot of the scrutiny and necessary QC that I spoke about as far as the challenges associated with spatial biology. But certainly we have done our due diligence in characterizing the most appropriate antibodies and done that needed QC, aspects of which that my colleague Harry actually spoke about. So again, these are ready to go for your trial needs. I do want to highlight a few. Clearly during the talk today, we showcased some of the really wonderful images using what we call our TME panel. So that's the third panel down. It includes 16 markers, and you just see some of the classifications that we can pull out of that. Clearly T and B cells, of course, you can delineate out your M1, M2 macrophages. Of course, thinking about, proliferation status of the tumor, that's always important. And again, I just highlight a few of the other well-known marker compositions, but I'll draw your attention to that final panel, which is the TIL-TAM vessel panel. There's a lot of interest, especially in some of the ongoing clinical trials today with PD-1 and VEGF. This would be a fantastic panel to use to further characterize, again, efficacy of that particular trial. And so, again, I do want to highlight, though, it's not just about off the shelf. We can tailor any marker to your specific needs and create that as part of your own dedicated panel. So we have custom approaches. Again, that's using perhaps maybe our validated assay with maybe slotting in one or two dedicated specific markers for your needs. Or we can do a full custom, again, using some of the quality and some of the rigor that we've applied that I spoke about during the talk today. So again, full custom, that's absolutely de novo assay development, and we can accommodate all of these approaches. And so I'll end with just touching on some points. I know that we've talked about this as we've gone through the webinar today. Again, we're really excited to be launching our Paletrra workflow. Again, it's really about that comprehensive AI-driven spatial proteomics approach, really to enable, it's about that whole tissue and being able to tell that story of the whole tissue to gain much deeper insights into what's going on within the tumor microenvironment. Again, calling out some of maybe the challenges associated with spatial biology, which is really in some of the pre-analytical variables and some of the QC. It's about the robustness of this particular assay. We have now offered specific panels that, again, have a very reliable assay performance. So it's about ensuring that we have robust end-to-end solutions for our clients. Again, really what sets us apart and what my colleague Harry alluded to is that advanced AI-driven image analysis. Really, it's about deconvoluting some of that complexity into kind of meaningful insights. And really how we do that, it's about the integration of a very dedicated team of pathologists and scientists on hand that really have that expertise to quantify and characterize the spatial arrangement, not just of proteins. We didn't really get to talk about RNA markers today, but certainly if there's any interest in integrating, for example, ISH probes and RNA, that's something that we can absolutely accommodate. And we certainly have posters that we highlighted during this year's AACR to discuss aspects of that. So again, with regard to Paletrra, it can be both off the shelf and we offer custom capabilities. And it's really about empowering you guys with more confident biomarker development and really achieving some of those clinically meaningful outcomes. So I really appreciate your time on the webinar today. I look forward to some further discussion in our Q&A session. So thanks very much for your attention, everybody.
Thank you both for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just type the question into the Ask a Question box and click Submit. We'll answer as many of your questions as we have time for. Let's go ahead and get started. Our first question asks, what equipment or instruments do you need for this? And can you combine this with hybridization? And does the cell quality suffer? What is the price?
Great question. I can take that. By the way, can everyone hear me? Yes. So I just want to make sure that we're all clear that the Paletrra workflow that we just went through, this is a full service offering, really, from the due diligence with qualifying antibodies and our very comprehensive down selection process, all the way through to image analysis. And this is exclusively available at NeoGenomics. We have applied very specific AI-driven algorithms that, of course, my colleague Harry went through, as well as applied the due diligence and some of the considerations with quality. Again, this is exclusive to NeoGenomics as a full service offering. So again, if there's any additional concerns with that approach, I'm more than happy to take that question if you want to clarify. Again, this is available only at NeoGenomics.
Excellent, thank you.
Yeah, and if I can add to that question as well, in terms of the in situ hybridization, we do incorporate RNAscope into our workflows, and again, as Kirsty said, we carefully ensure that all of our antibodies are compatible with this integrated RNAscope protein workflow.
Excellent, thank you. All right, our next question here. This question asks, what are the main challenges in implementing Paletrra into clinical trial workflows, and how is NeoGenomics addressing them? Also, what AI or visualization tools are used to communicate insights from such complex spatial data?
That's quite a question. Okay, I'll start with the first part of it. And again, I think it's quite clear that I think we all should be thinking about spatial biology and indeed, of course, the use of our Paletrra workflow to really get more understanding of your disease process, but also importantly, to really understand therapeutic responses. Because really, let's be honest, the immune system is tricky. Long-term response with various therapeutics, I think, has been a problem in the field. And really a lot of that is driven by immune exhaustion. So with Paletrra, of course, we're able to adequately address that. So first and foremost, I think anybody that has worked in any aspect of spatial biology would agree it's truly informative to include, especially in early phase trial. And of course, I think some of the publications that I highlighted really did a nice job of kind of addressing that and, applied some of the context as to why, with some therapeutics, we see some regression. And, there was, of course, that nice paper from BioNTech earlier this year. So, I'm a strong advocate for including multimodality approaches into your clinical trial, but especially including that much needed context as far as, understanding precise location and some of the interactions. Again, better understanding of that as part of your clinical trial. And again, I'll hand over to my colleague, Harry, who can address some of the AI aspects to that question.
Yes, so one key aspect of our AI pipeline is that we have integrated it very heavily with the imaging and staining workflow, and so it's all sort of integrated end to end and very tailored to the types of image outputs that we generate. And so these are all developed in-house and really uniquely designed to deliver the highest quality of data that we can for our modality. In terms of sort of helping our clients visualize some of these complex spatial insights, we rely on the Omero software ecosystem to allow our clients to interact with image data as well as overlay some of the analytical data in sort of an interactive browser-based tool. And then also we generate. So all of our analyses are essentially tailored to specific projects, tailored to a specific panel, tailored to specific research questions. And so we will generate visualizations to support addressing those questions.
Excellent, thank you. All right, moving along here, our next question is asking, is analytical software available for purchase or download, or is it only offered as a service?
The software is part of our end-to-end service and not available for download. It's not a standalone software.
All right, thank you. Okay, moving on. Our next question is asking, how can Paletrra improve validation of protein biomarkers in tissue when evaluating clinical utility of compounds of interest?
Yeah, I think we addressed some of that actually in question 2. And I will reiterate, it's all about, and of course, I would strongly recommend a multimodality approach to validation of your specific biomarker. I mean, we have to see beyond. As much as I think there's a lot of value in IHC, and especially IHC even for patient stratification purposes, we have to see beyond that. It's about making sure that that particular biomarker is meeting its right location. Is it interacting with the correct cells? And does it interact with the correct cells within the complexity of that overall tumor microenvironment? So again, I'm a strong advocate for including spatial biology technologies within the realm of your early phase trials. Again, having just come back from ASCO last week, and indeed even AACR back in April of this year, I think immune exhaustion remains, really a key consideration in, efficacy of many therapeutics, especially even ADCs and others. And so we really have to find improved ways to adequately address that. And, using Paletrra and using our full service workflow for that, I think really accommodates some of the need in that arena. I hope that addressed the question.
And if I can add a few comments as well. Thanks for the great answer, Kirsteen. In addition, I can just provide a little bit of information of sort of what we do at the antibody level and panel level when designing our assays. So the antibody level, we're really down selecting the best clone possible for detecting a protein of interest. And as part of adding that antibody into our test menu, we're working with our pathologist to identify the correct positive and negative controls. And all of our antibodies are signed off by our pathologist to ensure that they're demonstrating correct sensitivity and specificity within tumor tissues. And so in addition, once we build that out into a panel, we will then perform further qualification that all of the antibodies behave correctly within the full panel, and we'll follow that with fit for purpose validation. And so all of our work is really tailored to our clients' needs, and so we can provide the necessary level of validation of that panel that our clients require.
Wonderful, thank you. All right, next question. If I were to design my own panel for Paletrra, what are the main considerations I need to take into account?
So there's a couple of things, and there's a couple of very easy things off the bat. So first and foremost, we've got an extensive list of verified markers, using that validation process that Harry just alluded to. So, one can choose from over 200 markers that we have already extensively verified within our portfolio. So I would certainly, if that's a first step, then look at our extensive list for that, and I'm more than happy to share that, and certainly reach out to us as well. If it's a novel marker, of course, that's something that we can absolutely bring in and customize and develop. And again, I think Carrie did a great job alluding to our overall process for not just overall procurement of some of these specific clones, but our very comprehensive down selection process as far as qualifying these antibodies. And then, of course, we would certainly perform, the singleplex concordance as well as multiplex concordance in that regard. So again, I really think what sets us apart is that due diligence up front, especially with our overall QC process.
Wonderful, thank you. All right, moving along, we have a few more questions left in the Q&A inbox. Again, if you have questions that you'd like to ask our speakers, please do so now. Our next question is asking, what is the best way to quantify and interpret the spatial relationships between different cell types within a tissue sample, and which metrics are most useful for understanding these interactions?
Yeah, so I can take this one. So the answer is going to be that this really is something that needs to be tailored to a specific research project. And that's why our analysis pipeline is very flexible to support generating multiple different readouts depending on the research questions. And so I think really at a high level, when looking at spatial relationships, you need to understand some concept of interaction range, whether that's sort of close by cell-cell interactions or longer distance interactions, such as from cytokine secretion. And so, really the interplay between the abundance of cell types of interest, the interaction distance, and all of that sort of mixed in with the heterogeneity of the tissue itself and the sort of distribution of tumor cells. All of that together will really guide how to really develop the appropriate spatial analysis.
Okay, excellent. Our next question here says, typically, many image analysis tools are greatly influenced by inconsistent staining results. How does software account for inter and intra-run variability in staining quality?
Yeah, so I think first off, we deal with this by reducing inter and intra-run variability as much as possible. That's always the best solution, to not fix things with software, but sort of go to the source. And that's where having an end-to-end service is really great, because we control all aspects of this process, from the staining and imaging all the way through to the analysis. And one of the ways that we do this is by having a robust phase-in process for every lot of antibody that we run, and then also coupling this with necessary validation of all of the equipment within our CAP-CLIA lab to ensure minimal staining and imaging variability. Downstream, when we come to the actual analysis, it's very important that we have our batch controls. So this allows us to ensure that the staining is comparable between different batches of analysis. And then furthermore, by using really pathologist-guided AI tools that interpret both the intensity of the staining plus the morphology and staining patterns, that allows us greater robustness to various tissue-specific artifacts that might pose a challenge with more simple rule-based classification methods.
All right, wonderful. Next question that we have says, with the enormous variability of shapes and sizes of cells coupled with the variable expression of each of the biomarkers in individual cells within different labeled cell populations, How does your image analysis software account for this?
Yes, so we have been developing our AI pipeline since 2017, so about 8 years now. And so we have a wealth of annotated data across a large number of cell types, tumor indications, body sites, that allows us to train these AI algorithms that are really both specific to our assay, but also robust to the wide variety of tissue and cell types that we come across through our clinical trial testing.
Okay, wonderful. We have a couple more questions that we have time for, so let's move right along. Our next one is asking how your AI-driven image analysis is different than other solutions available in the market.
Harry's getting all the questions today.
Yes.
Yes.
So I would say there are quite a few AI tools available now. And so some of the ways that we differentiate ourselves, one is ours are custom built for our assay, and that improves the accuracy of some of our models compared to, for instance, pre-trained off-the-shelf models that might have been trained across a variety of different assays. And in addition to the sort of algorithmic accuracy that we've spent a lot of time on, that really builds upon the large, expertly annotated single-cell data that we've collected over 20 million individually annotated cells across all of our biomarkers. In addition to that, due to our experience in performing AI-driven image analysis over the past 8 years, we have a very robust QC process to ensure that these algorithms are performing as expected and we're generating the highest quality data possible.
All right, thank you. And we have time for one more question today. This last question is asking what level of validation is done to ensure the quality of the antibodies used by NeoGenomics, and how are the combinations of these ABs tested for your panels that you indicated?
Yeah, so I think we've, well, on you go.
Yeah, we've touched on this, but I can go on a little bit more detail as well. So, so as I mentioned, it begins with single biomarkers. We work with our pathologists to get them signed off, ensure that they're staining the correct cell populations and they're really performing as expected based off of our intended use of those biomarkers. We do additional characterization to understand really how to best build a panel of these antibodies to ensure the optimal performance of all of our antibodies. So then we once we build a panel, and this process is fairly robust. We also have a wealth of experience in doing this. So, you know, as Kirsteen mentioned, we have off-the-shelf panels. We also support customization of these panels. And so designing these custom panels is something that we have a lot of experience in, and typically a very high success rate in getting our, you know, translating from a single biomarker that shows the correct staining sensitivity and specificity to then having that biomarker work within our panel. And typically it's a very quick development process as well. And so once we have that full panel, we will run that on a set of samples to ensure that, again, within the full panel, we're seeing appropriate staining. And we will follow that up with fit-for-purpose validation that may include accuracy against gold standard, precision, such as repeatability and reproducibility of the panels. And so we can really add on the necessary levels of validation required by our clients.
I couldn't have said it any better. Thank you.
Perfect. Okay, I want to thank you both for your time today and your important research. We would also like to thank LabRoots and our sponsor for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions submitted during the live and on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.